Now moving towards the renewal activity of the gingival epithelium. So basically the oral epithelium undergoes continuous renewal. So okay, and its thickness is maintained by the means how the thickness is maintained, that the new cell formation occur in the basal, then it moves to the superficial layer. Okay, the spinous layers, and then the shedding of the old cells at the surface occurs. Okay, this activity shows in 24 hours periodicity with the highest and lowest rates which occurring in the morning evening. The mitotic rate is higher in non-keratinized areas and increase in gingivitis. Okay, without any gender differences. Basically, there are some that mitotic activity is increased or decreased with age. Okay, with regard to junction epithelium, so basically that epithelial cells facing the external basal lamina are very rapidly dividing, means the only epithelial cells that is facing external basal lamina are fastly divide. However, the evidence indicates that the significant number of cells are capable of synthesizing, means they are saying that the big number of cells produce DNA by their mitotic activity. The rapid shedding of cells effectively removes the bacteria. They are saying that rapidly remover of the cells, that if or rapidly shedding of the cells basically removes the bacteria that adhere to the epithelial cells, and therefore this is an important part of the antimicrobial defense mechanism at the dento-gingival junction. So they are saying that the important antimicrobial defense mechanism which is present at the dento-gingival junction is basically rapid shedding of cells, due to which there is a removal of the bacteria. Sometimes if people ask you that can we brush our gingiva, so there is no need to brush your gingiva, because the gingiva has also a very rapid shedding of cells. The new cells are formed and the old cells are removed. With that, the bacteria is also removed. Okay, so very important thing. Okay, now moving towards the cortical structures on the tooth. So basically what is cortical? Cortical are basically a thin acellular structures with a homogeneous matrix that is sometimes enclosed within a clearly demarcated linear bodies. So they are saying these are the acellular structures and they have a homogeneous matrix that is sometimes enclosed within a clearly demarcated linear borders. Okay, they are also acquired coatings. In acquired coatings we include the exogenous origin, such as we have saliva, which is actually exogenous bacterial calculus stains. Coatings of developmental origin are those that are normally found as a part of the tooth development. This includes reduced enamel epithelium, coronal cementum, and dental cortical. Okay, when the enamel formation is completed, so basically the ameloblastic epithelium is reduced in number of cells, number of cell layers basically that remain attached to the enamel surface by hemodesmosomes and lamina propria, basal lamina. The reduced enamel epithelium has also a post-secretory ameloblast and consist and cells from the stratum intermedia of the enamel origin. Okay, it's very important. Okay, so the connective tissue that deposits a thin layer of cementum is known as coronal cementum on enamel. The coronal cementum is basically a connective tissue cell layer that is a very thin layer of the cementum. Okay, and afibrillar cementum may be seen in the cervical half of the crown. Okay, basically. Okay, then they are saying that these cortical structures have a thickness of around 0.25 micrometer over the enamel surface. Okay, in a it is non-mineralized and it is not always present. In some cases, it is also near the cemento-enamel junction deposited over a layer of the afibrillar cementum, which is turn overlays enamel. It is deposited over sometimes over afibrillar cementum. Okay, it is also present between the junction epithelium and the tooth. Okay, dental cortical is a proteinaceous and it may be accumulation of tissue fluid of it is basically maybe a accumulation of tissue fluid components. Okay, now moving towards the gingival fluid. What is present in your gingival fluid actually? Either it can be a transudate or exudate. It contains biochemical factors, thereby offering its potential to use a diagnostic or prognostic biomarker of the biologic state of the periodontal in health and disease. Okay, it contains also connective tissue, epithelium cells, very important, inflammatory cells, serum, microbial flora that inhibit the gingival margins or sulcus, means that does not allow the bacteria to invade. Okay, so what is it has connective tissue, epithelium, inflammatory cells, serum, and microflora. Okay, in healthy sulcus, the amount of gingival it is very important that in the any inflammation of the gums, the gingival fluid is more. But here, why it is more? Because there are more inflammatory cells will be present. Okay, but in healthy, it is always less. During inflammation, they are saying gingival fluid increases and it resembles to an inflammatory exudate. Okay, the main route of gingival fluid diffusion is through the basement membrane. Okay, so what they are saying, that the main route of the gingival fluid is through the basement membrane, that is relatively wide intracellular spaces of the junction epithelium. So we have studied that the junction epithelium has it is porous, so the fluid can move in and out of that and into the sulcus. Okay, then the gingival fluid is believed to have following, these are like cleansing material from the sulcus. It is the functions. Okay, very important. It cleanses the material from the sulcus. Okay, it contains plasma proteins. They may improve the cohesion, means adhesion of the epithelium to the tooth. Very important point. Okay, and antimicrobial properties also as it. Okay, then it shows antibody activity to defend gingiva. So it has four functions. Number one is cleansing, number two is adhesion, antimicrobial activities, and also antibody activity to defend the gingiva. Okay.